hello again, everybody. Long time no see. Okay, so I think I'm on the agenda today to be talking about Wnt signaling, but I've kind of shifted it, and I'm going to break this up into two mini talks. So first, I'm going to talk, especially talking after Wendy Chung, I wanted to sort of give an overview of the natural history data that we've been able to gather through Citizen, and then I'm going to completely shift into giving a brief touch on Wnt signaling. So CTNNB Connect and Cure, I just pulled this from the website. I'm sure this is probably actually updated since this has been written, but we've got over 430 families globally, which is just excellent. At COMBINEDBrain, we work with a lot of really rare genetic disorders, many of which have far fewer than you guys. You've been really good at patient recruitment and finding patient recruitment and just patient engagement as well. So this kind of goes into the same as with Simon Searchlight, but the benefits of having an online registry and natural history study is that there's less caregiver burden. So a lot of natural history studies are conducted at a clinical center where patients go in and actually see doctors, get assessments, get blood draws, take surveys in the clinic itself. But the benefits of a lot of these online platforms that are really just taking off, especially in this day and age, there's no travel required. These studies can be global, so it's available to people from all different countries, ethnicities, backgrounds, often even just different like socioeconomic backgrounds, where often just cost of travel is very burdensome. And you can actually use all of this data aggregated to cross-validate other natural history studies with each other and other researchers. And it can really inform, especially for the foundation, for us it can inform where we can base future in-person studies and future in-person events. We can locate where are the people so we can go to you. Okay, so I'm going to talk about, let me pull up my pointer. I'm a pointing type of person. So we've been working with Citizen Health. This is their little logo here to collect data from all of the families. So as of currently, there are over 100 different patients registered in Citizen, and I'm going to talk today about the number of curated participants. So Citizen is really good at pulling in your EHR data, which is your electronic health record data. So you can link your electronic health records to Citizen, and it's going to pull in all of that data that your clinicians have in their systems, and then Citizen itself, their team, works very hard to curate that data. So they go through all of these, often they're even just grainy PDFs, and go through them and turn it into actual data that can be analyzed. So there are 40 patients that I was able to get data for very recently. This is actually kind of hot off the press in terms of us pulling this data. 40 patients that had enough data to really be curated and had consented to get their further data to be used. So the average age was actually very similar to what Wendy was saying. So about 8 years old is our average, with a range of 3 to 23 years old. And the average time since that person had last logged in, or their family rather, had logged in and updated it, the average was about 2 years. If you kind of look at where these people are on the map, it really reflects actually the map I showed 2 slides earlier with people who have registered on the website, so that's good to see. I kind of just said this, but here are some graphs showing the age of patients. So it looks like, again, an average of 8 years, but we have patients going, from a very young age up to, I think our oldest was almost 25 years old. What I found interesting was age at diagnosis. I'd be curious actually to look at this by diagnosis date too, because we had this one outlier of a person who was diagnosed at 16, when most people, which is great to hear, are diagnosed at a much younger age. So genetic report findings. This was actually really good to see a confirmation that because CTNNB1 syndrome is haploinsufficiency, so it's lack of beta-catenin, or at least a dysfunctional beta-catenin. So we see the majority of these are frame shift or nonsense mutations. When nonsense mutation is when you have a premature stop of the protein, which then normally you have kind of this half-formed protein that will go and get degraded. Frame shift mutations often lead to a premature stop as well, and that's what I saw. There were a few missense variants. It'd be interesting to really investigate. I come from a background of investigating missense variants, so exactly how those affect protein function, and a couple intronic variants. And I was actually a little bit personally surprised is that, maybe it matches up with the type of people who are going to sign up for Citizen, but these are majority classified as pathogenic variants. I've seen in other rare disorders where a lot of variants, if you actually just do like a disease prediction model, a lot of the variants, there's just not enough information for our different databases to really accurately predict that. So this is actually fairly strong for a genetic disease of this type. Also, an interesting thing was for genetic findings, about 3/4 of patients had just CTNNB1 mutated, but a good quarter of people had other genes as well. The top genes that I saw were these ones here. Some of them are of interest were that, oh no, my mouse. There we go. I'd like to point to MFSD2A. That's a protein involved in the blood-brain barrier. And we also have, what is it? It is CERKL is associated with retinal problems, retinal dystrophies. So it's interesting how we're seeing genes that are correlating with a lot of the same symptoms as classical CTNNB1 symptoms popping up as being co-mutated. And I looked also at chromosome, and none of these are you would assume maybe they're right next to CTNNB1 on the chromosome, which is why they're co-mutated, but that didn't really seem to be the case. So what was interesting is we looked at number of clinical diagnoses per patient. These are diagnoses like global developmental delay, hypotonia, ADHD. Patients really range in the number of diagnoses that we were able to pull from their electronic health record data, anywhere from just a couple of diagnoses to over 100 of them. And this is a summary of the top most frequently reported diagnoses. So global developmental delay, gait, hypotonia, constipation, microcephaly. A lot of these are very familiar with all of the talks we've seen earlier today. And if you just compare this side by side with the primary disease domains that you see in the literature, this is pulled from the Gene Reviews article for CTNNB1, you see really strong correlation. Although I really wanted to point out constipation here as a very commonly reported thing that you were seeing in the health record data that isn't perhaps as frequently reported in the literature. So that is my summary of kind of what we've been able to look at from Citizen Health. I would welcome any recommendations for how to really further dig into this data. This was kind of the base pack of data that we were able to download, but there is more data available that we can pull from, or sort of which other studies we really should start comparing this data to. How can we really help each other and not reinvent the wheel with this? Okay, now I will switch to Wnt signaling, which is kind of my my current hyperfixation. So the canonical Wnt signaling pathway, I already gave this talk for those of you who I already gave this slide for those of you who maybe just came in recently. So the canonical Wnt signaling pathway, beta-catenin really is the canonical Wnt signaling pathway protein. Maybe other Wnt signaling folks will be offended by that, but from my eyes, it really is. So normally this is in every cell of your body from the beginning of when you're a zygote through till the day you die. Every cell of your body has Wnt signaling more or less active. So in an inactive state, beta-catenin protein, which is what CTNNB1 encodes for, it gets phosphorylated by this complex of proteins that's called the destruction complex. And when beta-catenin gets phosphorylated, it gets degraded by the proteasome, which is kind of the, I call it like the garbage disposal of the cell. It just kind of shreds proteins that do not need to be used and then recycles the amino acids. But when you have this Wnt, it's a very small little signaling molecule, Wnt, that binds to this transmembrane frizzled receptor. This actually recruits the destruction complex to itself, which preserves beta-catenin from getting phosphorylated and consequently degraded. So then beta-catenin is free to do its job, which is to translocate into the nucleus, where your DNA is, and to interact with this family of transcription factors to upregulate Wnt signaling genes. So Wnt is really, really important for just global health and development. So there's kind of this what's called a Goldilocks paradigm, where too much or too little can cause consequences. So too much or too little Wnt signaling during development can cause developmental disorders in different ways. So for CTNNB1 syndrome, we're looking at a Wnt beta-catenin loss of function. We have loss of beta-catenin. But really gain or loss of function can cause different disorders, often very much associated with macrocephaly or microcephaly, language delays, severe intellectual disability, a lot of neuronal problems with early differentiation and development and maturation of neuronal populations in the brain and throughout the entire body. So the more I looked into this, so again, I work for COMBINEDBrain. We work with, I think, over 110 different rare disorders, such as CTNNB1. And the more I did a literature dive, and the more I saw that there are a lot of these rare genetic disorders that impact really foundational proteins for growth and development that sort of converge on the Wnt signaling pathway. So I started sort of collecting these. Sunny in the audience, she did a really great job helping me collect a lot of this, just do a big literature dive and kind of collecting all the different papers that connect these different disorders to Wnt signaling. And so from them, I sort of pulled together the foundations that were interested in maybe investigating this topic further. And so we've created this kind of working group that meets every couple of months, and we just put our heads together, and we're trying to collect the science, collect the data, and see if there's common therapeutics we could potentially work towards. Here's a list of the therapeutics we've discussed in our working group call. I wish I had sort of a diagram showing where each of these target, but we've been looking at Wnt mimetics, so something that will bind to frizzled. We've been looking at Norrin, it's a protein that interacts. We've been looking at inhibiting Wnt inhibitors. We've been looking at disrupting beta-catenin, GSK3 inhibitors. Michelle Jacob is going to give a great talk on that in a few minutes here. And then other small molecules and compounds, anything that's just already a supplement on the shelves at the drugstore to drugs that may be FDA approved but are not yet really commonly in use. Last slide, basically. So our goals of the Wnt working group are to, again, understand the biology of how Wnt affects our different disorders, discuss and identify common Wnt pathway targets for therapeutic rescue, find the researchers in the field who have that knowledge base, see what groups, communities they're plugged into, bring them to our cause, and really see if we could start investigating this as a subject in and of itself. We're recruiting basic scientists, clinicians, and foundation leaders who want to contribute to these efforts. And I'm really excited to see where this goes. This was kind of just like a little, I don't know, brainchild I had, and I'd like to see where we go with this. Also, in correlation with citizen data, we're throwing so many QR codes at you guys today. Thank you for your patience with us. But here's another QR code. This one just leads to the CTNNB1 research page, which then leads to all of the other QR codes to sign up for all the wonderful studies that are going on. So thank you, everybody.
Testing. And this is another QR code. This is the conference hub. It's also in your welcome packet. It has all the resources. You can pull it up on your phone if you lose your folder or anything. We just wanted to make sure you had it accessible as well. And now we're going to do a quick Q&A session for Dr. Chung and Kellan. If you could hang out. I'll start with the first. Yeah, you can go up there. Sorry, Wendy. And Kellan, do you mind just hang out up near the mic, because we have these two mics, and I'll run around for questions. But I'll start with the first question. We heard about Dr. Chung, the Simons and your clinical natural history study, and we heard from Kellan the citizen natural history study, which is completely digital. So I want the families in the room to understand how those work together and how, can you talk about how those work together and paint the picture?
Yeah. So I do think they're overlapping information, but complementary. So what I mean by that, and we've made ways that with your permission, we can connect the dots. That is that we can put the same information with your permission together, and then as researchers use it, of course, use it de-identified. What I mean by that is that there are certain things that we as doctors don't do a good job of documenting in the medical records, and I think you as parents, you know. And so that's some of the things that we can get from Simon Searchlight that aren't in the medical record. On the other hand, sometimes it's hard to remember, like you rack your brain in terms of when did that first happen. The nice thing about the medical record is it's time-stamped, that we have a sense of, at least from documenting what your providers heard from you, they've time-stamped it in terms of going forward. And sometimes they're medical terms or laboratory terms or other things that are more granular than either you'd remember or you'd care about. So I think they're complementary in terms of doing that. And I think as others are trying to think about clinical trials, both from a safety point of view, what do we need to be careful about, as well as what do we think about what we can measure and what makes a difference. All of those things come together in terms of understanding the natural history. I don't know if you want to say anything else.
You covered it all. Okay. I can say no more.
Thank you. Any questions in the audience? We'll run. Yeah.
Hello, so I have a son who's 9 years old with CTNNB1 mutation. So my question is about the pre-adolescent stage, about is there like beta-catenin being active? I know that there's probably a lot of brain development for teenagers, and I was wondering if maybe we might have missed the critical pathway when they're in utero, but is there a possibility that if there are therapeutics that it would help for someone in the preteen years? Thank you.
So I'm going to speculate, but I'll actually open it up to anyone else in terms of thinking about this. So I'm saying this in a more broad way. So it's a critical and fundamental question that you've asked, is what is the window of opportunity in terms of for a therapy to be able to have an effect? I actually think the therapy that we've learned from starting with Rett syndrome, Angelman syndrome, other things, that window is open longer than we think. I don't think we know until we know. And we can use animal models to be able to help understand this, and I think they are helpful, but animal models aren't people either, and so ultimately it's going to require people to be able to answer your question. I will say across many conditions, what I've realized is that many of these genes are important both for development and for maintenance of the brain and function. And one of the things, as I alluded to, I don't think we know what the lifespan looks like when people are in their 30s, 40s, 50s, and 60s. I know from other conditions that I started out studying young people with, and then eventually went from the other end of the spectrum and realized that we needed those genes for continued maintenance over life. And so I just think it's important to think about the entire lifespan and not give up on anyone as we're going forward. And we'll know when we know, because we'll do the experiments and then we'll see. Michelle may want to answer.
Oh, yeah.
There's good evidence, actually, that that pathway is important in every stage of life all the way through, and then it plays a role in plasticity and learning. So there's reason, I think, to be optimistic that you could get really good outcomes at any later stage of treatment as well. You don't have to hit it really early.
Thank you. And if you could introduce yourself as well.
My name is Stephanie. My 2-year-old son has CTNNB1. It's our first conference. So my question is, when you talk about the WNT work group, as a parent, I know I'm not a researcher, I'm not a clinician, but a lot of times I interact with a lot of clinicians. Is there any recommendation for like my child's providers that I could give them or their organization to help be a part of this WNT work group?
Yeah, I mean, if this is something that your clinician, if you mention it to them and they express interest, we'd love to have them. I think we're at a stage where, we're looking at a lot of basic science, but also trying to think how can we approach this, towards therapeutics. But yeah, if this is something you want to mention, if you think they have an interest, send them my way, please.
Email her.
Yes. Yeah.
Hey, Josh Rash, son, 2 years old, with CTNNB1, obviously. question for you guys, not necessarily anything that you've presented on, but more so for us to take home. Are all these presentations going to be available for us to like digest? Like, I mean, it's really fast, right? It's like Fast and Furious, and it's really small, and I'm sure a lot of us would prefer that. I just haven't seen that for you guys. Maybe there's a way we can put it on a Google Docs or something for everybody to look at.
Yeah, all these presentations are being recorded and will be available online afterwards.
Cool. I'm not talking about a recording. I'm talking about like the presentation themselves. Like that way, you're not like the slides.
Yes.
Sorry, different jargon. You know. Yeah, slides. But that's all I wanted to ask. Okay, cool.
Hi, I'm Beth, and my 12, almost 13-year-old daughter has CTNNB1, Libby. And thank you for everything. Honestly, you guys are amazing. This is our first conference, and my wondering is, we see her neurologist yearly, and she has her upcoming checkup coming up in September. What would be the top things that I should take away from your work that I can bring to him back in Grand Rapids, Michigan? What can I tell him or suggest to him as we move forward? Thank you.
So I'd say, and I'm saying not just for neurology, but where your daughter is in terms of her life stage. In the checklist of things, if someone hasn't evaluated, so if the ophthalmologist, if she hasn't seen the ophthalmologist recently, go back to the ophthalmologist. For her pediatrician to check for scoliosis, given that she's going through growth stage, I think that's important. With puberty, everyone, is going to be hormonally changing during that time as well, and so you might see some changes in behavior. I hope not changes in epilepsy, but for your neurologist specifically, you can take back this information. We're not seeing it, but again, we've got some limitations in terms of how many adolescents that we're seeing. And then if you're seeing anything, and I don't mean to single her out, but based on any symptoms she's having, seizures, autism, anything else, be happy to talk with any of us. But you can bring back the slides and actually, hand the deck of slides and say, this is what I learned. Do you see anything on this list that you're worried about? Tethered cord, have you checked for that? Are you concerned about any of the things you see on this list?
I can be fast.
So we have a 6-year-old daughter with CTNNB1, and in the last 6 to 8 months, we had our geneticist doctor approach us about therapy for Rett syndrome. And since you mentioned that, I was just curious if you're familiar with the debut brand and your opinions on that. We've been on it now for about 6 months, and we've seen some good progress with our daughter in the ways that helps. And I really hadn't been able to talk to anyone that specializes in this about it. So they specialize more in Rett, and they have several patients on it. So we're the first ones for them. So there's been some articles, I believe, that connect. If I didn't know my daughter had CTNNB1, I would have thought she had Rett syndrome. So.
I have to say, this is the first I've heard in terms of anyone with CTNNB1. I've got other patients with Rett syndrome that are on the medication, and there are, because some of you have younger children, just be careful. There is a side effect that comes with diarrhea with that medication, so I don't know that's for everyone.
You're well away on the plane ride, too.
But I'm glad anecdotally, I think one of the things as a community is to understand what people have done, learn from what they've done, trying to take away, what are the gains and losses in terms of as people experience these things. But I haven't heard of anyone else on it, so.
We've had really good results so far, if you're curious to talk.
That's great.
Thank you.
Is that a medication?
I'll come back.
Hi, I'm Anita Garza. I have Mia, and she's 18. So I know we're one of the older ones. And I just wondered, thinking about, this might be a stupid question, but the surveys for the, family history and all of that, how often do you want, would it be beneficial to redo those? Because I'm just realizing, we've done so much with the transition to adulthood, and I have a lot to share, but I didn't think about going back on there and updating, so it's sounding like it's really important. And also, would there be any reason to go back and ask for an updated genome test as well if it had been over 9 years, or is there any time, because it changes so much, would there be any benefit to do genetic testing again after you have a diagnosis? Thanks.
I'll say very briefly, on an annual basis, we try and make it light so that it's not too burdensome, but I do think it's helpful while it's fresh in your mind to be accurate for that reporting. But as you said, there are particular ages which are probably more things are going on, either because society is changing around you or because your body is changing. So, but that's the method to the madness. In terms of reanalysis, you had mentioned variants of uncertain significance. Most of you don't have those. This is a very easy gene to interpret the variants for, but if you do have a variant of uncertain significance, I do think that's worthwhile going back and getting reassessed. Very rarely it happens, but that someone's in the wrong group, and you actually need to find out this is not your home, but that something else is. But again, this gene is very easy to interpret. For the most part, that's going to be the main driver in terms of the brain and behavior. There aren't other genes that are coming into play, but there could be, if you've got a family history of something completely different, heart disease, breast cancer, something else, that's a completely different reason, but a reason to get that information if you want to know that as, just in general, the same as you would do with your own health and trying to manage your own health, but not specifically, I think, for neurobehavioral issues.
Can I just say for citizen data too, sometimes it's good if you kind of make sure and if you log in maybe also probably annually to make sure your EHR data is being pulled in accurately, because sometimes it can disconnect or, maybe it hasn't auto-updated. So at least just checking in on that to make sure that's up to date as well. And any other surveys that may be on Citizen, we would love to have.